Afternoon and welcome to the second special session, part of the Personalized Medicine Conference. Today we'll discuss about cancer genomics. The event is part of the European Biotech Week, and as I mentioned, part of the fifth edition of Personalized Medicine Conference, an event organized by Center for Innovation in Medicine. I'm Marius, the founder of the center. And we have a strong partnership with Europa Bio, with Local American Working Group, and Foundation Medicine and Roche. Thank you all for joining us. I'm sure that we'll have a great session on cancer genomics. Our special guest for today is Dr. Benedikt Westphalen. Benedikt is the head of Molecular Diagnostic and Therapy Program and the Molecular Tumor Board at Comprehensive Cancer Center, University of Munich in Germany. As I mentioned, we'll discuss with Benedikt about comprehensive genomic profiling, or in other words, about cancer genomics, precision medicine in oncology, personalized medicine in oncology, as a reality for patients and for medical doctor. Thank you so much, Benedikt, for being with us today. Now I invite you to have your lecture, and then we'll have the chance to interact. So feel free to use Q&A or the chat section from Zoom to address questions or to share your thoughts with Benedikt. Thank you so much. Please.
Yeah, hello from my side. Thank you very much for the kind introduction and the invitation to speak today. My name is Ben Westphalen. I'm a medical oncologist and molecular biologist. And we've been running a precision oncology program here in Munich. We're now in the 5th year. And I would like to discuss precision oncology in clinical practice, but also chances and challenges with you today. And I hope we will be able to have a great discussion later on, because there is a lot to discuss. These are my conflicts of interest. So when I think about precision oncology, I always think a little bit about a puzzle around genes, around oncogenes, and we have almost all pieces to the puzzle readily at hand, but now it's time and our duty to fit them together to deliver the full potential of precision oncology to our patients. And sticking with this idea of a puzzle, I would like to discuss briefly the concept of precision oncology, precision cancer medicine, the evidence, and maybe trouble that we have with evidence at the very moment, the medical reality with a couple of examples, and then how the future might look and what we can all do in order to fully implement precision oncology into clinical practice. So in terms of the concept, why should we aim for broad usage of comprehensive genomic profiling in cancer medicine? The idea being, and this is based on some of the precision medicine glossary of ESMO, is that you have a great group of cancer patients, and then if you do comprehensive molecular profiling, you will be able to divide those patients into groups, and this informs not only clinical practice, but this is also very important in terms of enrollment into modern clinical trials and drug development, as you have classic companion diagnostics. Let's say we look for HER2 or RAS. You are able to find biomarkers that will allow your patients to enroll into clinical trials. And then also, and we cannot forget that the data that is generated can help, if we know more about cancer, can help to define targets for drug development, research and development in general. So the first concept, or the first statement I would like to make, that genomic profiling really guides therapeutic management these days. These are data from the MSKCC Impact program, one of the largest precision oncology programs in the world. And yes, this is based in Upper Manhattan, a very wealthy hospital, but it should illustrate you that patients that undergo molecular profiling at the beginning of their patient career have a very high chance to be enrolled into molecularly guided clinical trials. And this is really an important example where I also see a lot of potential. And it was mentioned that you are in collaboration with a company providing genomic profiling. Um, Even in countries where you don't have broad access in the general population, if you have specialized centers investing in this technique, maybe partnering with academic partners, you will be able to enroll patients into clinical trials and give them access to drug very early on. And looking at the paper trail coming from MSKCC Impact these days, almost every other week you see large clinical studies with novel therapeutics out there. So this is really to illustrate that not only for your daily practice, but also for providing access to care, comprehensive genomic profiling is really important. One of the issues that has always been discussed over the last couple of years is genomic profiling too challenging and too slow for our patients. And you see data from questionnaires, and you have in green patients, and this should actually be what we aim for. And the question was, if you are able to undergo comprehensive genomic profiling, would you be willing to delay treatment? And 2/3 of patients said, yes, I would wait for the results to come in. Almost 70% of patients said, yes, sure, I would go for another biopsy if this is informative for my treatment. And almost 90% or more than 90% of the patients would spend their remaining tissue to the hospital. So it's not that patients don't want to participate. We just have to explain them what we're doing, and then they're willing to go this extra step. And then on the right-hand side, you see that over 60% of patients are willing to wait 2 weeks or longer when it comes for the results. And I mean, here in Munich, profiling, variant annotation, plus discussion in the molecular tumor board takes around 4 to 5 weeks. And this is one thing that we can discuss later on, when the optimal timing would be to aim for a comprehensive genomic profiling and maybe discussion in a molecular tumor board. So the implementation or the implications of comprehensive genomic profiling in clinical practice. We see more and more profiling being used. Costs are decreasing and turnaround times are shortening. If you have enough case volume, you can run the whole testing thing within a week easily. And comprehensive genomic profiling is really the driver behind precision oncology in clinical practice. Think about non-small cell lung cancer, how many molecular targets we see. Think about biliary tract cancers, where you now have a lot of new targets emerging. Then, and this is really important for me, if you have access to comprehensive genomic profiling, you will enable yourself to run modern clinical trials and allow access to innovative drugs to your patients. And one problem that we will have to discuss in this setting is that you will often face the situation where you don't have approved drugs at hand, and then we enter the off-label realm. This is certainly a topic that we can discuss later on. So now we come to a place that is a little bit more difficult. And when I talk about evidence, I would like to talk about the use of comprehensive genomic profiling to drive precision medicine in the individual cancer patient, not for just non-small lung cancer, but in the typical setting, a late-stage cancer patient coming to your practice, asking for a diagnostic test. Where's the evidence that this really helps our patients? And there was this great editorial in Cancer Research a couple of years ago that I highly recommend reading. So, are we vaguely right pushing precision oncology, or precisely wrong because it's not the right road to go down. And one of the problems that is always brought up is that we don't have good clinical trials to support the broad usage of comprehensive genomic profiling driving targeted treatments in advanced cancer patients. But we need this evidence because we have more precise diagnostics, more precise drugs. Think about NTRK inhibitors, RET inhibitors, but the classic clinical trials that we ran back in the days with 500 patients being randomized, that's just not possible. And there is a lot of debate going on. Is it ethical for NTRK inhibitors, for example, to run randomized clinical trials because the response rates are so good and the patients are so rare that it is almost unethical? So this new precision oncology really defies the principles of classical oncology trial making. Nevertheless, we need evidence because we as doctors, the first thing we have to keep in mind is do no harm. And we need to justify decisions in front of our patients, in front of regulators, and also in front of payers. We cannot simply ask and say, well, we believe precision oncology is working. Give us as much testing as possible. We will play around with off-label drugs and you let us be. That's not how doctors and oncologists should run their clinical practice. And how is it possible to then generate evidence? So, and this is why I really like this multi-stakeholder approach that you're running here. You need all people on the table. This cannot be solved by doctors alone. This cannot be solved by payers, insurance companies. You need policy, you need industry, you need drug makers, you need doctors, you need patients. All of them should work together to generate the evidence needed. We need smart and innovative trial designs, and I will briefly touch on novel data sources. So just very briefly, is there evidence? It has been tried in broad clinical trials to actually look at the use of comprehensive genomic profiling and a molecular tumor board in advanced cancer. And the most prominent trial is the Shiva trial by Christophe Le Tourneau from 2015, which did not show any benefit of molecularly targeted agents when compared to treatment at physician's choice. However, you have to remember, back in the days, this trial was run in the early 2010s. We didn't have the drugs, and we didn't have access to drugs to really make the fullest potential. And Shiva 2 is running, and I'm really excited to see what comes out there. The first trial that showed that there is a role for molecular tumor boards and precision oncology, in my opinion, is Moscato, where patients were used as their own control. So you did your comprehensive genomic profiling, had a molecular tumor board, and if you found a targetable alteration in this trial, patients and their progression-free survival were compared between the last standard chemotherapy or last treatment and your experimental treatment. And you can see that there was a good amount of patients that really benefited, where PFS2 was far longer than PFS1. Here in red is PFS1 and green is PFS2. So the ratio for the targeted agent really being good. One of the learnings from Moscato was, and this was a second paper, is that patients with biliary tract cancers especially benefited from these targeted agents. And this is a theme that we see more and more. So this is great work by Razel Kurzrock from UCSF and their colleagues. What they did is they did a huge meta-analysis of phase 2 clinical trials based on biomarkers. And what you can see is that response rates, median progression-free survival, and median overall survival are all longer if you go for a personalized approach in these molecularly guided trials. So there is a signal, and it is up to us now to really tease out where precision oncology based on comprehensive genomic profiling will make the biggest impact. One thing that then automatically comes out of this, and I really want to hammer on this point, in such specialized and N of 1 cases where you have a molecular tumor board and you treat a patient, it will be very difficult to do this routinely in clinical trials. So whenever we treat, we need to document what we're doing. So this is where real-world data then becomes big. And real-world data that was called 1.0 really does not have the detail and the standards that we can use it for anything that could be considered evidence in the end. On the other hand, clinical trials, very narrow scope, only fit patients, one metastasis, running a marathon back and forth from the clinic. So this is really not transferable into clinical practice then. So the idea was, and this was really a great presentation, ASCO 2018, where from the NCI in America, there was this educational session that really stated maybe real-world evidence collection, if done right, is supplementing and maybe even replacing clinical trials in certain settings. And the idea was that you had real world 2.0. You still had the broad scope. You had your young patients, you had your old patients, you had patients from all different areas in your country. It was still fragmented, but you increase the level of detail, you increase the level of quality, and you standardize. And then this data becomes really powerful. And I believe this is a dialogue that when you have these multi-stakeholder discussions, how to implement precision medicine on a national level, such as you are aiming for, real world evidence, in my opinion, needs to be implemented here so that we or you guys actually know what comes out in the end. What is the reality? The reality looks actually better from year to year, I have to say. So personalizing cancer care is not a dream anymore. When we look at data from ASCO 2018, the Taylor X trial, showing in a huge trial of more than, I believe it was 6,000 patients, 6,500 patients, if an RNA-based 21 gene recurrence score can digest women out that do not benefit from additional chemotherapy versus endocrine therapy alone in hormone receptor positive, HER2 negative, node negative breast cancer. And this was a non-inferiority trial. And if you move very close to your monitor, your screen, you can see that there are actually two lines because it was essentially the same here. So now using a 21 gene recurrence score, you are able to prevent women from toxic chemotherapy and just treat them with endocrine. And this is really personalized precision oncology on a large scale. Secondly, we all saw the first histology agnostic approval of pembrolizumab across malignancies in MSI cancers, and now also, as of recently, in cancers with a tumor mutational burden of greater than 10. This was really championed in the USA. Unfortunately, EMA has not followed the lead, and it's really interesting to see if we will see histology agnostic approvals of this scope in Europe as well. We've seen NTRK inhibitors. The next group of agents will most probably be the RET inhibitors, but MSI high cancer is still a big gap here in Europe, I would say. And another theme that really supports now precision oncology and shows us how important it is to implement comprehensive genomic profiling into clinical practice are these rare alterations that you will find across tumor types. So we spoke about TRK fusions, larotrectinib and entrectinib, now EMA and FDA approved, or RET fusions with pralsetinib and Loxo 292 selcatab. now I don't know how to pronounce, but LOXO 292 also FDA approved. And these markers with amazing clinical activity, you won't find them by IHC, you won't find them by a single PCR, you will only find them when you do more extensive profiling in the end. And this theme, we've seen a lot of data presented at AACR, ASCO, and ESMO. We cannot not see this being reality in the next 2 to 5 years. What's the future? I briefly alluded on these rare targets where you have patients that will derive benefit from all these novel therapeutics. And the necessity here is really for cancer patients to allow them to be diagnosed, that you find these rare alterations, and you won't find them if you screen just one patient. If you have for NTRK a prevalence of 0.3% in adults, you won't find it if you screen every once in a while. You need really access to testing to then find those patients that have these rare alterations, but also to drive standard of care, as I said, in lung cancer, in biliary tract cancer, even in colorectal cancer. If you do MSI testing, RAS and RAF testing, you have 3 things that you could run on a small panel, which will be more cost effective and less tissue wasting when you compare it to IHC and 3 or 4 PCRs that you need to run. The near future, and this is a theme that you all know, is that we need molecular tumor boards to really work with the evidence that we have, allocate patients to clinical trials, and to discuss these results of comprehensive genomic profiling to offer therapeutic guidance in the end. And I'm happy to discuss the Munich model later on. Our vision is, and this is what we're still trying to build, integrative molecular oncology or precision oncology, where you have your specialized center, you offer the chance for outpatients and inpatients to come. You have a diagnostics core, you offer therapeutics, clinical trials, and also a molecular tumor board and a virtual molecular tumor board to work with your referral centers and other doctors, so that this is not an ivory tower where you keep for yourself and do not really share your knowledge, but that you spread the word that precision oncology is here and it's here to stay. There are a lot of known problems, and I will go back to this slide maybe later on. What's the indication? We need to work on education. We still need to define what molecular targets are in diseases not well covered. What is a molecular tumor board? How do we ensure access to treatment? We have spoken about real world evidence, and I put it, the biggest problem I put down below. This is all costly. I wouldn't say expensive, it's costly, but we need to come up with idea how this can really be implemented into clinical care. And with this, I would like to conclude that omics-driven precision cancer medicine is clinical reality. We cannot discuss this away. But medical reality really needs to catch up. And this is not only the thing in Romania, it's also a thing in Germany. We really need to figure out how we do the best for our patients. This starts with medical education. There are a lot of naysayers and nihilists and pessimists still saying that this will never work. We need to catch up in terms of access to testing and drugs. We need to get better in trial design and data generation. Drug approval has to follow, and drug design will also change in the near future. And the vision really would be that we all together, and that's why I really like your approach, work together across sectors to really build a better future for our patients. And with this, I'm done here and happy to discuss with you.
Thank you so much, Benedict. Feel free to ask questions, use the chat or Q&A for this, or share your thoughts. I like entire lecture, but especially the vision, the idea of co-creation for patients and together with the patients. And this was also one of the conclusion of the plenary session that we had earlier today on the Personalized Medicine Conference. Indeed, personalizing cancer care is not a dream anymore, and it's a reality. I don't know how to start actually, because we have a lot of challenges, we have a lot of problems, a lot of opportunities. You already mentioned some of them, but I think it's very clear from the beginning. You mentioned that we have to have in mind the evidence, and I think it's important to somehow to clarify what are the type of cancer, or I don't know, clinical entities that who can benefit more from.
Mm-hmm. Yeah, I completely agree, and I mean, this evidence problem maybe stems from the time where comprehensive genomic profiling was seen as a technique of last resort. You know, when we started the molecular tumor board 4 years ago, or 5 years ago, the idea was patients that had no options left would undergo profiling, and we hoped to find a silver bullet. And this we have failed miserably. So this doesn't really work. If you try to save a patient life at the very end when the cancer is very advanced and resistant to everything. So and in these days, people would always say, well, A, you are not able to show any benefit. B, your patients are dying anyway, and it's too slow. And there I see this connection to real world evidence, really. So, I mean, we have now profiled roughly 1,500 patients in our program. And now we begin to understand exactly what you were asking for. Which patients do really benefit from comprehensive genomic profiling? Where should we maybe go more aggressive, maybe in first or second line? And the best example, I have to say, I mentioned it a couple of times, are biliary tract cancers. Those are cancers with a dismal prognosis, but now we have a lot of very promising targets that you can also already address. And this is how medicine becomes a self-learning system, so that you really see what works in my practice, what works with my friends and colleagues, and then say, okay, if we did it 10 times and it doesn't work, it won't work the 11th and 15th time. And if you put all of this together, as I said, you're growing and you're creating the evidence needed, and you're not wasting a lot of money and creating hopes that are not working out well.
You are working in a comprehensive cancer center.
Mm-hmm.
One of the potential recommendation actually for the EU cancer plan, that every member state to have a number of comprehensive cancer centers.
Mm-hmm.
And I've seen some the idea of molecular tumor board and virtual tumor board.
Yeah.
And I would like to explain your model from Munich. Mm-hmm. What exactly means? What are the type of specialists involved? How actually you work there?
That's a good question, because we there is really no good definition of a molecular tumor board. So what we in Munich found very helpful is that you always have the primary care team at site. So it doesn't make sense to just look at a foundation medicine or in-house report and say, oh, this is really interesting, let's try this or that. You need to hear firsthand from the primary care team how the patient is doing, what past medical history he or she has. This is, like, for me, the core. Secondly, it helps to have a good molecular pathology core, so people that really are invested in the techniques and know how to what also the pitfalls are. We have also a geneticist in there, because more and more we see hints for germline mutations when doing the tumor-only testing. And then it depends, neuropathology. So I would I would say the most important thing is that you have molecular pathologists, the primary care team, and then a dedicated team of people that really start building their own experience, educating themselves, building knowledge. What is a targetable alteration? What is working? What is not working? I always say, with a little bit of a smile, precision oncology, when you want to run a molecular tumor board, is not a hobby. You cannot do this on the side if you really want to keep up with the immense speed that this has now developed. You would just be missing a lot of opportunities in the end.
You mentioned the in-house test you are doing in Munich, and say commercial test, foundation type of test. And I think it's important also to explain what is the medical thinking for a certain patient. I don't know, we can imagine a non-small cell lung cancer patient maybe, and a breast cancer patient. And what do you recommend first? What is the sequence of using a type of test or another type of test?
Yeah, that's a very good question. So I mean, we are in the lucky position that in Germany you can order the test and the insurance company pays for it. So there is reimbursement. So you're really free to use any test that you would want to use in a given situation. For example, when TMB really came up, we didn't have the in-house capacity to test for TMB. So in all cases where we thought TMB might be an important target, we sent for out-of-house testing, essentially. Yet, on the other hand, the beauty of having in-house testing with pathology is we have different assays. We use a lot of Oncomine, which is relatively cheap and covers a lot of known targets. It has its weaknesses, but it works with very little amount of tissue. So especially when you mention lung, for example, where you have very, very tiny pieces, sometimes more comprehensive tests such as Foundation Medicine, but also TSO 500 use up too much tissue. So this is really great if you in-house can play around. You know, in the end, I would have to say that it doesn't really matter where you get your testing from, unless no, it doesn't matter where you get your testing from, as long as it's quality controlled and you trust your testing. So if you trust your molecular pathologist, and I do, then I'm happy to send most of the cases to him. If you don't have in-house testing, a company like Foundation Medicine with 400,000 cases diagnosed all around the globe, they better know what they're doing, and they are highly efficient in providing you with good test results. You then still need someone, and this is my a very important take-home message for me. You still need someone who really looks at these results, because even a Foundation Medicine test is sometimes hard to interpret. Sometimes it's a little bit misleading. So you need people that actually look at the case, at the testing, say, well, this is interesting. Here we have an opportunity, or, well, this looks interesting at first, but we don't think that it's going to work therapeutically. So it really depends on what you have at hand, and if you are able to work with the results that come out in the end.
And a very tough question, but what would you do if you don't have actually anything in your hands? In other words, these tests are not reimbursed. You don't have an in-house test. Maybe you don't have experts.
Yeah. I mean,
but also
yeah.
Tumor board.
I sort of expected the question because you briefed me beforehand a little bit. So yeah, that's a good question. So I mean, again, I see multi-stakeholder cooperations being the key here. And if you are in a middle income country and reimbursement of testing is not done, what I would try to do is really come up with a plan to, let's say, you do testing for diseases where you really know it makes a difference. So set up a lung cancer testing program together with a maybe with a company, negotiate with your health Department of Health, so that you say, okay, we in Romania want to show that it's feasible and that we actually deliver to our patients, and start learning in easy waters, so for lung you will have a lot of success stories in the end, and you learn how to work with these reports, and you have a good use case. In the end, you can also build your real world database, which then in turn is interesting for maybe the company that is supporting you, or for your national cancer registry. It's also important for your payers to show that you actually do good for your patients with the testing. Because if you look at the whole costs a cancer patient causes, the testing, let it be 4,000 euros if it's expensive, is a tiny fraction of what a cancer patient actually costs. Yes, I know. If you then go for targeted drugs are also expensive. But if you have someone with an ALK fusion living 3 or 4 years without evidence of disease, this person can go back to work, pay taxes, be productive, and so it refinances in the end. So yeah. My guess would be try to build a small program, learn from others, ask people that have already made a lot of mistakes. That's why I find it great that you invited someone from Germany. So building this together is really a good thing to start. And I mean, happy to help and consult, because I've done it for colleagues in Italy, for colleagues in the US. Why not Romania? Would be actually fun.
Yes, let me tell you a story. Actually,
Yeah.
2 years ago at the Personalized Medicine Conference, we did what you actually already explained. We put all the stakeholders together for a program on non-small cell lung cancer. The idea was to have a national protocol for testing the biomarkers. At that time, if I remember correctly, were 4 or 5 mandatory biomarkers. But nothing happened. We had a consensus of all pharma companies. We discussed with all the key opinion leaders and decision makers. And now we have to actually rewrite the white paper, because we have 10 or 11 mandatory biomarkers, and some of them NTRK and other in relation with tumor agnostic drugs. So you are welcome to the white paper on non-small cell lung cancer 2.0. Actually, we'll start working on this, and this time hopefully we'll succeed. To convince the national fund, insurance fund, and the authorities to implement this kind of program. Let's continue with our discussion. We have also some questions from the audience.
Yeah.
You mentioned the TMB, and Beatrice asked, what are the lessons learned from your journey with TMB in your clinic? What are the do's and don'ts?
So in the beginning, I was a strong believer in tumor mutational burden. And I have to say that in non-MSI or non-POL-E mutant cancers that usually have a very high TMB, our experiences with TMB high cancers that we treated with immunotherapeutics are not overwhelmingly good, I have to say. I mean, looking at the it's Keynote 158, I believe, that just came out, looking at the histology agnostic approval for Pembro. The data is okay, but I would say it's not the most important biomarker that I would consider having in my portfolio. I would, if I had to choose, and for example have a good test to look for fusion events, for example, I would rather go for fusions than have TMB in it, to be quite honest, because the number of patients that are MSI or microsatellite stable, TMB high is low. And yeah, as I said, at least in my personal opinion or experience, it was not that great.
And we have one comment and one question from Astratine. The comment is regarding the patient registries. We have an issue in Romania with this topic. And Violeta is saying, unfortunately, real data collection would highly depend on good patient registries, which is not the case for Romania. And the question for you, Ben, is could you speak a bit more about your research program, which programs are critical in precision medicine program, and what is the role and the contribution?
Yeah, thanks, Violeta. So briefly answering to the first statement, I mean, you could easily build in your practice your own registry to start with. I mean, there are good white papers on what needs to be collected, what does not need to be collected. And this is actually very valuable data where you could start with your own institutional or multi-institutional approach, just coming up with the idea, how do we collect data from clinical practice? It doesn't have to be a fancy internet-based thing where you automatically shift data back and forth. It can start slow. So at least that's what I believe. But it's important to start collecting if you have these interesting cohorts. So in terms of the research program, so it is, in my opinion, absolutely critical that you link your comprehensive genomic profiling / molecular tumor board to an early clinical trials unit, or at least build knowledge and a small program where you have a couple of trials that actually stand behind your tumor board. The best tumor board and the best profiling is useless, as we discussed, if you don't have access to drugs. Also in Germany, it's difficult to get off-label approval. We have to write to the insurance companies, so forth and so on. So, and this should not be the aim of a molecular tumor board. The aim of a molecular tumor board should be enrollment into a clinical trial. So this is the first structure that I believe is really critical. And this brings me to the partners that you would have there, in terms of that you build a good relationship with pharmaceutical companies that run these trials, so that once a trial comes, you are able to say, see, we can do the testing, we give you 5 patients a year, and then you're golden. This is the idea. And then you are automatically in some sort of a dialogue where it goes back and forth. So which partners are critical? Grant. We are supported by our Comprehensive Cancer Center. So this is state funding, actually. But we have industry collaborations as well, where we use third-party funding to support IT personnel and stuff like that. So offering drugs to be tested, yeah, that's, as I said, one of the biggest problems, and only a healthy clinical trial portfolio will prevent you from just running an off-label program, which also has some sort of need. We still prescribe off-label quite frequently, but it shouldn't be your primary goal.
It's clear in countries like Romania, low and middle income countries, that we have to start building a genomic profile, a genomic infrastructure for research and for clinical practice. This is clear. You mentioned the education, and I would like to ask you more on how to actually to persuade and to convince the medical doctors and to oncologists to to be more proactive, to more open to this kind of new technologies.
Yeah, so I believe that the only way to do that is, I mean, in some ways you have it easy starting a little bit later because you build on, I always say there is a mountain of shitty data. 10 years ago, everyone said, we just have to test cancer and we're going to cure it. I'm just exaggerating. But now look at what happened this year, how many molecularly targeted drugs have been approved. So now and this is not just lung cancer, these drugs come into other diseases as well. And I believe that just very, yeah, calm and collected, showing that this can make a difference. It can make a huge difference in non-small cell lung cancer. But look at, for example, biliary tract cancers. I'm always bringing this up because this has been a cancer. When you think back 3 years, we had gemcitabine cisplatinum, and we had that since forever, and nothing has changed. And now you have IDH1 inhibitors, you have FGFR inhibitors, you have great data on BRAF targeting, on HER2 targeting. So this is really a poster child and an example where you can say, if we do it right, if we look for the right targets and the right disease, then we will be able to show you that it helps. We don't want to throw the testing on the market and everyone should get tested, especially if you have limited resources. We want to do it smart. Help us to do it smart and help us to convince you that the data that we collect, the patients that we treat will speak their own language in the end. But it's a tough way. We got a lot of hate here. We're told we were wasting a lot of money and time, essentially.
Yes, and discussing about money, you mentioned that actually the cost for test, it's a small part actually from the, let's say, total cost for cancer care. But using the comprehensive genomic profiling will drive us to and you mentioned the clinical trials as an objective for the molecular tumor board, let's say. Another issue is a member state has to provide timely access to approved drugs, and this is an issue, and there are differences between Germany and Romania. And the topic of label medication, and maybe I think it would be good for us to find more about what you mentioned a little bit. What is the case for off-label medication in Germany?
So in Germany, there is a famous court ruling called Santa Claus ruling, because it was issued on Christmas, essentially. And it says that if a patient has a life-threatening cancer or disease, you have had all standard therapeutic options exhausted, and the treatment that you want to prescribe has a reasonable chance, and a reasonable chance is most often a case report, essentially, then the insurance company needs to pay for it. It always goes first through the insurance company, then through a special agency that looks at all these letters from doctors, and then looking if this makes sense. So in theory, it's not that hard to then get reimbursement in the end. Still, it's forbidden to do this in a structured way. And so we're not allowed to, you're not allowed to really prospectively collect the data, because then it's off-label use in a structured way, as if we're running a clinical trial. So there is a gray area here. And I mean, we now try to be very defensive with our molecular tumor board in terms of only issuing statements based on national evidence grades, but also based on the SCAT from ESMO, that where we believe we can really make a difference for the patient. So a good example would be PI3 kinase mutations. Back in the days, people would always say, why don't you use everolimus or sirolimus? Those are great drugs in the indication where they are indicated, but it doesn't help to throw it onto a colorectal cancer or a pancreatic cancer in the end. It doesn't help. So you waste time, money, and you are doing no good. So we try to limit ourselves only to cases where we believe we can really make a difference for the patient. HER2 would be a good example. If you find a HER2 amplification or a BRAF mutation in cancers that are not breast or colorectal, usually working with drugs that target those pathways, you get a good response. And that would be my advice, that you only take high priority targets and not waste your own time on non-good targets.
We have a few more minutes, and I would like to somehow to provoke you.
Yeah,
we
go ahead.
With Eric Green about personalized prevention and polygenic scores. Also, in the morning, we discuss about this concept on personalization of the prevention. How do you see the role of genomics for cancer prevention?
Yeah, that's a good question, actually. So I'm clearly not an expert in this field, but I would say from my educated guess, there's so much fascinating data looking at, you name it, gastric cancer in Asia, and people moving for there's great data from Chinese families moving from China to the US, and their kids, first generation Chinese in America, have the same risk as Americans, while their parents still have the risk growing up in China. So there is a huge potential to find susceptible subgroups, but this would require so much more efforts to really go into large-scale screening programs, simultaneously looking at activities of daily living, food intake, so forth and so on. So I believe we will be better and better to find out high-risk groups. I mean, we're good with BRCA families or other cancer families. So I see in high-risk individuals, I guess we're going to get better and better. Moving this to a more global scale, difficult. My feeling, but clearly this is just an educated guess because I'm not really a prevention person. Oh yeah, just one thing, Violetta, you're right. Your name came. It was I remembered that I had seen it before, that we met in Brussels, and I agree that we need to take patients and their advocates along. Absolutely.
Yes, Violetta is a very active patient advocate on melanoma, and we are very happy to have Violetta with us today. Maybe if you have other questions or comments for Ben, it's clear that we are actually at the beginning of the journey of implementation of personalized medicine and genomic medicine, and especially in oncology. I'd like to thank you so much, Ben, for your insights. It was very, inspirational, and I think you provide us some clarification on molecular tumor boards, on off-label medication. This is a very important topic. actually on the right use based on evidence for the use of comprehensive genomic profiling. And I would like to ask you a final question, because we have a lot of medical students and young doctors.
Mm-hmm.
What is actually your message? Because they'll go into the clinical practice probably in 10 years or in 5 years. What's your message? What they should expect in 5 or 10 years to find in a comprehensive cancer center here in Bucharest, in Cluj, in Iași, or other Romanian cities?
So my first thing would be to say that they should be really excited to enter clinical oncology these days. I mean, for me, I turned 40 this year, and I still have a good amount of time left, but still I feel that for younger doctors that are 5 or 10 years junior to me, they will have opportunities in personalized medicine that we were only dreaming of. I mean, we now see what is possible with these new with this interplay of profiling and new drugs. This is now really coming together, and this will not stop. So I believe that medical students, young medical doctors have a very, very interesting and fruitful future in front of them. But this comes also with an obligation. If you have such great tools at hand, you need to stay vigilant and use them right. So as I said, we're not in Germany, not in the US, not in Romania, in a position where money flows freely and we will be able to test everyone. So you need to be sure that you know where your tools are the most valuable to then make the maximum benefit for your patient. Yeah.
It's clear that, as you mentioned, and I repeat, the personalized medicine in cancer is not a dream anymore. It's a reality. And as you mentioned, it's just the beginning. It's just the beginning, and I'm sure that we'll have a very good future, especially in regards to cancer care, maybe to cancer prevention. And what is very important for us is to be able to set the right framework to make sure that we will take the value of the new advances in technology and genomics and data, what you mentioned. I really appreciate your model in Comprehensive Cancer Center. I follow the European Cancer Plan with the recommendation regarding this, and why not? Maybe we'll find ways to connect into the future and to somehow to translate your experience to Romanian oncology centers. Ben, thank you so much for that very inspiring our part of the Personalized Medicine Conference, and thank you all the attendees for participation.
Yeah, thank you very much also from my side. You can see I've put my email address and my Twitter handle into the chat. So if you want to DM me on Twitter or shoot me an email because you have other questions that you feel we didn't touch, happy to answer. And if at some point you're further along the road and I can be of any help or assistance, I'm happy to do that in any case, because as I said, we did a lot of mistakes, and others don't necessarily have to do them as well. So thank you very much. All the best to Romania.
Thank you so much, and have a great evening. Thanks.
Bye-bye.
Bye.